Assalamu alaikum everyone, and welcome back to a new lecture in renal physiology. This is a continuation of our previous discussion about reabsorption in different parts of the nephron in the kidney. Today we will discuss the reabsorption in the loop of Henle, and the discussion will mainly focus on the type of substances reabsorbed in the loop of Henle, the mechanisms involved in each case, and the consequences of dysfunction of the loop of Henle. So let's move to the drawing board and take a start. Here we have a schematic diagram showing the different parts of a nephron. The first part of the nephron, the proximal convoluted tubule, empties into the loop of Henle. The loop of Henle is U-shaped. It dips deeper into the medulla and it has got three important parts, each one having its own reabsorptive characteristics. The first part is the thin descending limb of the loop of Henle, followed by the thin ascending limb of the loop of Henle, and still later we have the thick ascending limb of the loop of Henle. The first part receives tubular filtrate from the proximal convoluted tubule, and as the name implies, this part, the thin descending limb of the loop of Henle, has got thin epithelial membranes without any brush border, and it has got fewer mitochondria because it has got less metabolic activity. This part is highly permeable to water, and it has some permeability for solutes like sodium and urea as well. However, water reabsorption in the thin descending limb of the loop of Henle outpaces that of sodium and urea. As a result, that is gradual concentration of the tubular filtrate as it passes through the thin descending limb. The thin descending limb receives the tubular filtrate from the proximal convoluted tubule that is isotonic. That is, it has roughly the same osmolarity as that of plasma, and it is usually about 300 milliosmoles per liter. As a result of water reabsorption and excess of solute reabsorption, the osmolarity of the tubular contents by the time it leaves the thin descending limb increases. The second part, that is the thin ascending limb of the loop of Henle, is virtually impermeable to water, and so is the thick ascending limb of the loop of Henle. However, it helps reabsorb some sodium from the tubular filtrate as it passes through this part. And as a result of some sodium reabsorption, the osmolarity of the tubular filtrate as it empties into the thick ascending limb of the loop of Henle slightly drops. The next part, the thick ascending limb of the loop of Henle, has got thicker membranes because it has to support a variety of active transport processes within this part. And for a better view, we must move on to a magnified view of the thick ascending limb of the loop. Well, here we have a schematic magnified view of a tubular epithelial cell from the thick ascending limb of the loop of Henle. Here on my left hand side, we have the tubular lumen that contains the tubular filtrate. Here we have a tubular epithelial cell for reference, and here on my right hand side, we have the renal interstitium. The side of the tubular epithelial cell that faces the tubular lumen is called the apical membrane, while the side that faces the renal interstitium is called the basolateral membrane. In general, about 25% of the filtered load of sodium, potassium, and chloride is reabsorbed in the loop of Henle, and most of this reabsorption occurs in the thick ascending limb. Like the proximal convoluted tubule, the reabsorption of everything else in the thick ascending limb is closely linked to the reabsorption of sodium, which in itself is closely linked or dependent on the activity of the sodium-potassium ATPase pump in the basolateral membrane. This pump continuously pumps sodium out of the cell in exchange of potassium that moves into the cell. So as a result, we get a buildup of potassium within the cell, and the intracellular stores of sodium are very low, therefore providing a favorable gradient for reabsorption of sodium from the tubular lumen into the tubular epithelial cell. Here perhaps I don't need to mention that the lumen contains the tubular filtrate, which is an ultrafiltrate of plasma and having a very high concentration of sodium. So once a concentration gradient has been achieved, sodium moves from the tubular lumen into the tubular epithelial cell along a concentration gradient or downhill transport. This downhill transport of sodium from the lumen into the tubular epithelial cell releases energy that is utilized to reabsorb potassium and chloride from the tubular lumen. So the sodium, potassium and chloride reabsorbed this way are in the second stage disposed into the interstitium. Sodium absorbed this way is pumped by the sodium-potassium ATPase into the interstitium. Chloride leaves the cell through specific chloride channels in the basolateral membrane, while potassium, which is in very high concentration within the cells, leaks into the interstitium as well as the tubular lumen through potassium channels. This is a characteristic of potassium channels that they are very leaky. So there is some big leak of potassium into the tubular lumen. So even though the sodium-potassium chloride co-transporter in the apical membrane reabsorbs equal amounts of positive and negative ions, the big leak of potassium through the potassium channels in the apical membrane causes a slightly positive charge of about 8 plus 8 millivolt in the tubular lumen as compared to the interstitium. This slightly positive charge within the tubular lumen causes the passage of calcium cations like calcium and magnesium into the renal interstitium through the paracellular route, that is through the spaces in between adjacent tubular epithelial cells. As we discussed earlier, the thick ascending limb of the loop of Henle is virtually impermeable to water. So it keeps on absorbing solutes like sodium, potassium, chloride, calcium and magnesium from the tubular lumen while leaving water behind in the tubular filtrate. As a result, by the time the tubular filtrate leaves the thick ascending limb of loop of Henle, its osmolarity is significantly lower. And by the time the tubular filtrate enters the next part of the nephron, that is the distal convoluted tubule, its osmolarity is as low as 100 milliosmoles per liter. So this is how the thick ascending limb dilutes the tubular contents. Well, it is quite logical at this point to ask, what if the thick ascending limb of the loop of Henle stops working? So one such scenario is the use of the loop diuretics like furosemide, bumetanide and ethacranic acid. This is a class of drugs used to treat edema associated with congestive cardiac failure, liver cirrhosis and chronic kidney diseases. The loop diuretics just block the apical sodium potassium 2 chloride co-transporter and these solutes are not reabsorbed. They stay in the tubular lumen and much of them is likely to be excreted in urine because the later segments of the nephron, the distal convoluted tubules and the collecting ducts have a reabsorptive capacity far less than the thick ascending limb. In addition, the presence of these solutes in the tubular lumen causes osmotic diuresis, that is loss of water in urine, thereby causing the risk of dehydration. At the same time, since the apical sodium potassium 2 chloride co-transporter extracts solutes from the tubular lumen and dumps it into the interstitium, it contributes significantly to the generation of a hyperosmotic renal medullary interstitium, a feature that is essential for the formation of concentrated urine. So blockade of the sodium potassium 2 chloride co-transporter also causes the loss of the renal urine concentrating ability. Barter syndrome is yet another similar condition that is functionally similar to the use of loop diuretics. Here we have a genetic mutation in the genes coding for this apical sodium potassium 2 chloride cotransporter, or the apical potassium channels, or the basolateral chloride channels. As a result, the dysfunction of these channels causes the subject to lose sodium and potassium in urine, and at the same time causes the risk of metabolic alkalosis. So what you need to know, loop diuretics and Barter syndrome are functionally similar, except that the use of loop diuretics is an acquired disorder and Barter syndrome is an inherited genetic disorder.
And that's it. This is the time to say goodbye to all. I know it was boring, but if you found it helpful in any way, please don't forget to share it with your friends, and thank you for your patience.